Okay, 13.4 is gene regulation and expression. So if you think of a library book filled with how-to books, would you ever need to use all of those books at the same time? Well, of course not. So now picture a tiny bacterium that contains more than 4,000 genes. Most of its genes code for proteins that do everything from building cell walls to breaking down food. So do you think E. coli uses all 4,000 plus volumes in its genetic library at the same time? So how are prokaryotic genes regulated? DNA binding proteins in prokaryotes regulate genes by controlling transcription. Bacteria and other prokaryotes do not need to transcribe all of their genes at the same time. So to conserve energy and resources, prokaryotes regulate their activities, producing only those genes necessary for the cell to function. An example, it would be wasteful for a bacterium to produce enzymes that are needed to make a molecule that is readily available from its environment. So by regulating gene expression, bacteria can respond to changes in their environment, the presence or absence of nutrients, for example. So DNA binding proteins in prokaryotes regulate genes by controlling transcription. And some of these regulatory proteins help switch genes on, while others turn genes off. The genes in bacteria are organized into operons. An operon is a group of genes that are regulated together, and the genes in an operon usually have related functions. So, for example, the 4,288 genes that code for proteins in E. coli include a cluster of 3 genes that must be turned on together before the bacterium can use the sugar lactose as a food. These 3 lactose genes in E. coli are called the lac operon. LACOPERON. Lactose is a compound made up of 2 simple sugars, galactose and glucose. And to use lactose for food, the bacterium must transport lactose across its cell membrane and then break the bond between the glucose and galactose. So these tasks are formed by proteins coded for by the genes of the lac operon. If the bacterium grows in a medium where lactose is the only food source, it must transcribe these genes and produce these proteins. But if grown on another food substance, such as glucose, it would have no need for these proteins. The lac genes are turned off by the proteins that bind the DNA and block transcription. So on one side of the operon's 3 genes are 2 regulatory regions. The first is a promoter, which is a site where RNA polymerase can bind to begin transcription. The other region is called the operator. This is where a DNA binding protein known as a lac repressor can bind to the DNA. So when lactose is not present, the lac repressor binds to the operator region, blocking the RNA polymerase from reaching the lac genes to begin transcription. So the binding of the repressor proteins switches the operon off by preventing the transcription of its genes. Okay, the lac repressor protein has a binding site for lactose. So when lactose is present, it attaches to the lac repressor and it changes the shape of the repressor protein in a way that causes it to fall off the operator. And when the repressor no longer bound to the operator site, RNA polymerase can bind to the promoter and transcribe the genes of the operon. So in the presence of lactose, the operon is automatically switched on. So how are genes regulated in eukaryotic cells? Well, by binding DNA sequences in the regulatory regions of eukaryotic genes, transcription factors control the expression of those genes. And one interesting feature of the typical eukaryotic gene is I would say TATA, but it's TATA box, a short region of DNA containing the sequence TATATA or TATAAA that is usually found just before a gene. So the TATA box binds a protein that helps position RNA polymerase by marking a point just before the beginning of a gene. And there's a really nice picture of that in your book. Gene expression in eukaryotic cells can be regulated at a number of levels. So DNA binding proteins known as transcription factors regulate gene expression at the transcription level. So by binding DNA sequences in the regulatory regions of the eukaryotic genes, transcription factors control the expression of those genes. So some transcription factors enhance transcription by opening up tightly packed chromatin. Others help attract RNA polymerase, and others block access to certain genes. But in most cases, multiple transcription factors must bind before RNA polymerase is able to attach to the promoter region and start transcription. Gene promoters have multiple binding sites for transcription factors, each of which can influence the transcription. Certain factors activate many genes at once, dramatically changing the patterns of gene expression in the cell. Other factors form only in response to chemical signals. Eukaryotic gene expression can also be regulated by many other factors, including the exit of messenger RNA molecules from the nucleus, the stability of messenger RNA, and even the breakdown of a gene's protein products. So why is gene regulation in eukaryotes more complex than prokaryotes? Well, cell specialization requires genetic specialization. Yet all of the cells in a multicellular organism carry the same genetic code in their nucleus. So complex gene regulation in eukaryotes is what makes specialization possible. So for years, biologists wondered why cells that contain lots of small RNA molecules, only a few dozen bases long, and don't belong to any of the major groups of RNA, they wondered why they contain lots of cells like the messenger RNA, the trans-RNA, the riboRNA. These small RNA molecules play a role in regulating gene expression by interfering with messenger RNA. So after they're produced by transcription, the small interfering RNA molecules fold into double strands, or what we would call like hairpin loops. And an enzyme called the dicer, which is just what it sounds like. It's an enzyme that cuts or dices these double strands into microRNA, each about 20 pairs in length. The two strands of the loops then separate. And one of the, one of the micro RNA pieces attaches to a cluster of proteins to form what is known as a silencing complex. And the silencing complex binds to and destroys messenger RNA containing a sequence that is complementary to the microRNA. So the microRNA sticks to certain messenger RNA molecules and stops them from passing on their protein-making instructions. So blocking gene expression by means of microRNA silencing complex is known as RNA interference. And at first, RNA interference seemed to be a rare event found only in a few plants and other species, but now it's clear that RNA interference is found throughout the living world and that it even plays a role in human growth and development. Okay, so let's see. The discovery of RNA interference has made it possible for researchers to switch genes off and on whenever they want to, at will, simply by inserting double-stranded RNA into cells. So the dicer enzymes then cut the RNA into the microRNA, which activates silencing complexes. So these complexes block the expression of gene production in messenger RNA, complementary to the microRNA. So the RNA interference technology is a powerful way to study gene expression in the laboratory. So it holds promise of allowing medical scientists to turn off and on the expressions of genes from viruses and cancer cells, and it may provide new ways to treat and perhaps even cure diseases. So that would be real handy right now with this COVID-19 out there. What controls the development of cells and tissues in multicellular organisms? The master control genes are like switches that trigger particular patterns of development and differentiation in cells and tissues. So regulating gene expression is especially important in shaping the way a multicellular organism develops. Each of the specialized cell types found in the adult originates from the same fertilized egg. So as an embryo develops, different sets of genes are regulated by transcription factors and repressors. And gene regulation helps cells undergo differentiation, becoming specialized in structure and function. So Edward B. Lewis was the first to show that a specific group of genes controls the identities of body parts in the embryo of the common fruit fly. Lewis found that a mutation in one of these genes actually resulted in a fly with a leg growing out of its head in place of an antenna. So from Lewis's work, it became clear that a set of master control genes known as homeotic genes regulate organs that develop in specific parts of the body. Molecular studies of homeotic genes show that they share a very similar 130 base DNA sequence, which was given the name homeobox. So homeobox genes code for transcription factors that activate other genes that are important in cell development and differentiation. And homeobox genes are expressed in certain regions of the body, and they determine factors like the presence of wings or legs. So in flies, a group of homeobox genes known as Hox genes are located side by side in a single cluster. And there's a picture of this in your book for you. Hox genes determine the identities of each segment of a fly body. They're arranged in the exact order in which they are expressed, from anterior to posterior. So in one of the figures in the book, the colored areas on the fly show the approximate body areas affected by genes of the corresponding colors. And then because it'll show you a fruit fly chromosome to an embryo to an adult fruit fly. And a mutation in one of these genes can completely change the organs that develop in specific parts of the body. So clusters of Hox genes exist in the DNA of other animals, including the mouse and humans. There's a picture of that in your book also. These genes are arranged in the same way from the head to the tail. The colored areas of the mouse show the approximate body areas affected by genes of the corresponding color. So the function of Hox genes in other animals seems to be almost the same as it is in the fruit flies. They tell the cells of the body how to differentiate as the body grows. So nearly all animals, from flies to mammals, share the same basic tools for building their different body parts, or different parts of the body. So master control genes, these are genes that control development, are like switches that they trigger particular patterns of development and differentiation in cells and tissues. So common patterns of genetic control exist between all these genes that have descended from the genes of common ancestors. So in prokaryotes and eukaryotes, and remember we're eukaryotes, environmental factors like temperature, salinity, that means the salt content, and the nutrient availability can influence gene expression. Pardon me. So, for example, the lac operant in E. coli is switched on only when lactose is the only food source in the bacterium's environment. Metamorphosis is another example of how organisms can modify gene expression in response to their environment. So, metamorphosis involves a series of transformations from one life stage to another, such as the transformation of a tadpole to an adult bullfrog. It's typically regulated by a number of external, which means environmental, and internal, which is hormonal factors. And organisms move from larva to adult stages. Their body cells differentiate to form new organs. And at the same time, old organs are lost through cell death. So under less than ideal conditions, a drying pond, a high density of predators, low amounts of foods, tadpoles may speed up their metamorphosis. The speed of metamorphosis is determined by various environmental changes that are translated into hormonal changes. So with the hormones functioning at the molecular level. Okay, that's the end of chapter 13.